Good evening, Asma. Good evening, Manchika. So, in the last class, we had talked about the development of ovule and the female gametophyte. We had talked about the integuments, chalaza, nucellus, the unitegmic and the bitegmic ovules, the micropyle end, chalaza end, and all that. So, I'm Rahul Saha, and today we'll be talking about the specific megasporogenesis process. So, before I commence with the megasporogenesis, I'll make you acquainted with two types of cell division. One is periclinal and another one is anticlinal cell division. Then we'll come on to megasporogenesis. So the periclinal cell division, okay, are the ones that occur periclinal cell division are the ones that occur, you can say, parallel to the tissue or organ surface and occurs mostly in plants. That occurs parallel to the tissue or organ surface. Meaning that if these are the two layers, this is the cell like this. So parallel means it will occur like this. The cell division will occur like this, you can say, like this. This will be the way the cell divisions will occur if this is parallel to the tissue surface. This is known as periclinal cell division. Okay. So as a result, we get rows of cells. What will you get? Rows of cells stacked on top of each other. So we will get rows of cells stacked on top of each other. Okay, so that is what you will be getting, or rows of cells stacked one over the other. Now, anticlinal cell division are perpendicular to the adjacent layer of cells. Once we talk about the anticlinal cell division, they occur, or it occurs perpendicular to the adjacent layer of cells. Layer of cells. So what you get is columns of cells instead of rows and stacks of cells on top of another, or one another. So what you get is columns of cells. We don't get the rows of cells that we were getting in case of periclinal division. Instead, we are getting columns of cells. So this is how the division would take place, like this. What you will get is columns of cells. Right? The columns of cells will be emanated from this kind of specific cell division. Okay, so in botany, based on the kind of divisions, like there are various kinds of plant meristems. We have got a rib or file when there is division along only one plane. Okay, either periclinal or anticlinal. We have got the plate weight division where there is division along both the planes. Again, periclinal as well as anticlinal. Then we have got the mass type division. When there is no fixed plane of division, there is only a mass of cells formed. So various types are there in botany. So that is what you should be knowing it. Okay. So now we will be commencing the specific megasporogenesis. So megasporogenesis, when we talk about, so in the nucellus, towards the micropylar end, there are specific archisporial cells in the nucellus. towards the micropylar end, okay, not the chalazial end, micropylar end, there are specific archisporial cells known as hypodermal distinct archisporial cells. Hypodermal distinct archisporial cell. Very, very important this is. Hypodermal distinct archisporial cell. So now these cells are very, very important. Why? Because they are the cells, archisporial cells, because they are the cells that divide by periclinal division. Again, that's why I explained to you the periclinal division. So they divide by periclinal division mitotically, okay, in a periclinal plane, resulting in an outer primary parietal cell and an inner primary sporogenous cell. So they will undergo mitosis. Number one, what kind of division? Periclinal division. Always remember for NEET examination. Periclinal division, and they'll form an outer layer of primary parietal cell, primary. You know that parietal means outer in our body also, primary and you can say visceral, right? So primary parietal cell plus inner layer of primary sporogenous cell. Sporogenous cell. So these two cells are formed after mitotic division. Keep in your head very, very clearly, it's a mitotic division, equational division, not the reductional or meiotic division. Okay. Now, primary parietal cell, it divides further. Divides means it differentiates further to form the parietal tissue of the nucellus. The outer one will form the parietal tissue of the nucellus. Now, this primary parietal cell will differentiate even further to form the, you can say, parietal tissue of the nucellus. Now, the primary sporogenous cell, now that will form the, or will differentiate to become megaspore mother cell. Okay. Now, primary sporogenous cell, keep in your head, it will again go differentiation. Differentiation means again mitosis only in both the cases. Okay. Here it doesn't mean meiosis. It means mitosis, and I'm writing it down so that you do not mess it up, right? Never get confused. Differentiation here means mitosis. Okay, never write meiosis. And then it will produce megaspore mother cell. Megaspore mother cell, MMC. When we write MMC, we really mean megaspore mother cell, not the microspore mother cell, right? That is a historical reason behind it. Okay. Now this megaspore mother cell is of diploid ploidy, right? Ploidy is diploid. 2n. Now this megaspore mother cell will now undergo meiosis. Okay. Will now undergo meiosis. So the thing is, The megaspore mother cell will now undergo, wait a minute, okay, it's done. Good evening, Tarun. So the megaspore mother cell, wait a minute, technical issue. Okay. Yeah, it's fine now. So the megaspore mother cell will now undergo meiosis. MMC will now undergo meiosis. Okay. When it undergoes meiosis, what it will form is, it will form 4 haploid megaspores, just like we had seen the microspore tetrad being formed in case of microsporogenesis. So 2N, again never forget the ploidy, it will form 4 haploid megaspores. Now 4 haploid megaspores have already been produced. This is the first step in megasporogenesis. Now listen very carefully, that out of these 4 megaspores in a linear tetrad, because they will also form a linear tetrad just like the microspore mother cells. In a linear tetrad, what happens is, usually the upper 3 megaspores, they degenerate. The upper 3 megaspores, they degenerate. Okay. When the upper 3 megaspores, they degenerate, what happens is only one will remain. Only one megaspore remains. The upper 3 degenerate. So write it down. The upper 3 megaspores, they usually have been seen to be degenerating or dying. Okay, degenerate. Only what happens is, the lowermost one enlarges to become the functional megaspore. The lowermost one megaspore enlarges to become functional megaspore. That is the case here. Okay. Now, after that, we will be talking about megasporogenesis with the topic known as development of female gametophyte. Okay. So we will take 2 topics together. Development of female gametophyte. Now, the functional megaspore forms female gametophyte or embryo sac. Okay. The functional megaspore forms the female gametophyte or embryo sac. Embryo sac here. There it was pollen grain, here it is embryo sac. So the nucleus of the megaspore, now it will start mitotic division. Once meiosis has happened, now again mitotic division would happen, just like the pollen grains, they had divided into one generative cell and one vegetative cell, and the generative cell divided again after it was landed in the stigma or the pollen tube has formed, or sometimes it was able to go through meiotic division inside the vegetative cell, but most of the time it undergoes the mitotic division inside the vegetative cell, but sometimes it can undergo mitotic division when it is mostly in the pollen tube in its journey towards the, you can say, stigma. Then what happens is, you have again mitotic division here, just like it is a symmetry. Microsporogenesis and megasporogenesis. Just learn these 2 topics as a symmetrical things. Try to correlate, and then both will be completely inculcated in your brain. So what happens is, you have the functional megaspore, the lowermost one which has become the functional megaspore, now that functional megaspore will do what? Will go for mitotic division. Okay, how many mitotic divisions? First it will become the functional megaspore will go for mitotic division. Now it divides into two. Okay, first mitosis 1, it will become 2 cells, right? Then again mitotic division number 2, now it has become 4 cells. Then again mitosis 3, it's become now 8 cells. All are haploid, right? Because this one is haploid. Okay. So what happens is it undergoes 3 mitotic division to form 2, 4 and finally 8 nuclei. It is not cells really, it is the nuclei. They are forming, the karyokinesis is happening, the cytokinesis is really not happened here. I just wrote cells so that it gets in your head, but really it is nuclei. Okay. Now what happens is 4 of this specific nuclei, they are divided into, you can say, group of 4. Now 4 nuclei each will be placed or will be located at each pole, okay, of the embryo sac. So 4 of them will be located at each pole located at each pole of the embryo sac. Okay. When they are located in each pole of the embryo sac, what happens is, now there will be a drastic event. The drastic event is 1 nucleus from each pole. Okay, 1 nucleus from each pole. Always remember, 1 nucleus from each pole will migrate. Okay, will migrate to the center to form 2 polar nuclei. Will migrate to the center to form 2 polar nuclei. Very, very important. Means 1 from each. Suppose you have got this 4 here. 1, 2, 3, 4. 1, 2, 3, 4. So 1 will migrate towards the center to form a polar nuclei. Okay. The polar nuclei will be formed and then they will fuse together. Okay. They will fuse together to form a diploid fusion or a secondary nucleus. So if you write it down, you can say 1 nucleus from each pole. From each pole will migrate or migrates to the center to form 2 polar nuclei. To form 2 polar nuclei, which will in turn fuse to produce a diploid fusion or secondary nucleus. You can call it a diploid fusion nucleus, or you can call it a secondary nucleus. And that is what it will do. Now what happens is the central cell now bears polar nuclei. Now what you have is you have 3 nuclei at the chelazal end of the embryo sac. Okay, they form the antipodal cells. The remaining 3 nuclei, which are present at now the micropylar end, they get surrounded by cytoplasm to form pyriform cells. Okay, these 3 cells are also known as, or also comprised of, the egg apparatus, which consists of 2 cells which are known as synergids or help cells, I should be saying, and an egg cell. The remaining one is the egg cell, which is present at the antipodal end, also known as the oosphere, which is present in between the 2 synergids. Okay, if we have this entire thing, okay, so this is the chelazal end. Okay, this is the micropylar end, if you see, basal end. So the 3 nuclei at the chelazal end of the embryo sac, they form the antipodal cells. They are known as antipodal cells. Okay, 3 nuclei at the chelazal end. In the middle you have got, you can say, a huge nuclei. This is a tetraploid nuclei, you can say, right? Not tetraploid, sorry, diploid nuclei. N and N has got mixed, so this is a diploid nuclei, 2N, because it has N and N mixed, okay, fused. Then at the, you can say, micropylar end, you have the 3 nuclei which get surrounded by cytoplasm, okay, to form the pyriform cells, okay, which constitute the egg apparatus. Out of those 2, these 2 cells and these 3 cells, okay, these are known by the name, you can say, synergids. Or help cells. And the middle one, okay, which hangs in between these 2, is known as an egg or oosphere. All have ploidy of N each. This also has ploidy of N each. This one is known as egg or oosphere. Very, very important. I can draw it with a different color. This one is the most vital, right? Egg or oosphere. So now we have been able to understand all the cells now. Now what happens is the synergids bear a special cellular thickening, or thickenings I should be saying, at the micropylar tip. They are known as the filiform apparatus. Okay, cellular thickening. They are known as the filiform apparatus. Very important, neat question. Okay, they play a very, very significant role in guiding the pollen tube into the synergid. Write it down. Guiding the filiform apparatus. They play a vital role, pivotal role in guiding the you can say pollen tube into the synergids, guiding the pollen tube into the synergids. So that is what they are most useful in. Now the egg cell, upon fertilization gives rise to zygote, while the synergids, what happens is they undergo or they get disorganized. Okay, the thing is they get disorganized very soon after the event of fertilization. Okay, after that, so the thing is fertilization means the generative cell plus the oosphere and or the egg cell, we call it, and the synergids, okay, or the help cells, they get disoriented, okay, or disorganized, I should be saying. They get disorganized soon after fertilization event. Disorganized soon after fertilization event. Okay. Now when they have got disorganized, this is followed by the disorganization of the antipodal cells. Now the antipodal cells also get disorganized. They also get disorganized. Now when the antipodal cells have also got disorganized, what it means is their function could be nutritive. Okay, still it is fundamental question is research, but their function could be nutritive. Okay, function could be nutritive. Okay, now what I can say is a typical angiospermic embryo sac, a typical angiospermic embryo sac can do what? At maturity, though it is 8 nucleate, is 7-celled. Very important, because of the fusion of the two cells, one coming from the chalazal end, one coming from the microphylar end at the center, forming the specialized secondary nucleus. Okay, so the developmental of embryo sac is from 1 megaspore only, because at 1 megaspore it gave rise to 4. That 1 megaspore, that functional 1 megaspore, okay, it divided 3 times via mitosis and gave rise to all these 8 cells, and that entirely developed this entire, you can say, embryo sac. Okay, so this functional, the development of embryo sac, I can say the development of the embryo sac is generally monospermic, that is from 1 megaspore. It's generally monosporic, I should be saying, monosporic in case of the angiosperm plants, right? So that is what happens. Know it very clearly. This is what we should be talking about, or we should be comprehending. So this is what happens, and on piercing the nucleus, what happens is, or on piercing the nucellus, I should be saying, on piercing the nucellus, the pollen tube penetrates the embryo sac. So after piercing the nucellus, the pollen tube penetrates the embryo sac and reaches the egg apparatus. It reaches the egg apparatus. When it does reach the egg apparatus, it passes either between the egg and synergids or between one synergid and wall of the embryo sac. What happens is, whatever happens, ultimately the tip of the pollen tube bursts and two male gametes are discharged, because that generative cell has again divided into two generative cells. So two male gametes are discharged. So tip of the pollen tube bursts and two male gametes are discharged. Now, two male gametes are discharged means we will have two fertilization event. That is why you might be getting the idea. We call it double fertilization. Okay. Now, what would happen is the tube nucleus disorganizes. It gets disorganized. The pollen tube or the tube nucleus. I said saying the tube nucleus means the nucleus of the tube cell or the vegetative cell. So the tube nucleus disorganizes. Okay, it disorganizes just before bursting of the pollen tube or prior to bursting of the pollen tube. Now, the one of the, you can say, male gametes, it fuses with the egg cell or oosphere. Okay, egg cell, one male gamete, right? I can say the generative cell, the male gamete, which has been divided into male gamete, plus the oosphere or egg cell. Okay, this is known as syngamy or generative fertilization. This event is known as syngamy or generative fertilization. Now, why is it known as generative fertilization? Is as a result of this fertilization, the consequence of this fertilization is the formation of a diploid oospore. This is N, both haploid. So the formation, the consequence is a diploid oospore or zygote. The functional zygote is produced. That is what is known as generative fertilization or syngamy. Okay. The other male gamete which I told you, which comes out, which emanates after the bursting of the pollen tube, after the disorganization of the tube nucleus, what it does is it goes and fuses with the already produced secondary nucleus which is present at the center. The other male gamete fuses with the other male gamete fuses with the, you can say, already produced secondary nucleus. Secondary nucleus, right? Now that fusion is known as triple fusion because the secondary nucleus is of ploidy 2n. Okay, the male gamete is of ploidy. You can say male gamete plus secondary nucleus 2n plus n. Okay. that leads to the formation of triple fusion or vegetative fusion, because you have ploidy which is now 3n. It is known as triple fusion. You can say two nucleus and one was three nuclei. So triple fusion, 3n ploidy again triple fusion. Okay, that is known as triple fusion, 3 nuclear fusion. You can say triple fusion. Also, it is known as vegetative fertilization. Okay, because it doesn't produce a functional zygote, so it is vegetative instead, so vegetative fertilization. Vegetative fertilization. This forms a triploid primary endosperm nucleus pen. The result is there it forms oospore or zygote. This forms primary endosperm nucleus. Primary endosperm nucleus, which has a ploidy of 3n, triploid, okay, which will later on become the functional endosperm. Which will later on become the functional endosperm. Now, this entire process of fertilization, which occurs twice in the same embryo sac, I can say, at one time, perpetuated by two different male gametes. So, two different events of fertilization, that is, one is triple fusion or vegetative fertilization combined with, you can say, generative fusion or syngamy. Okay, this is now these two fertilization, independent fertilization events forming primary endosperm nucleus and the oospore or zygote is now known as double fertilization event. So if you summarize it, it will be the oosphere plus male gamete. This is syngamy or generative fertilization. This gives rise to oosphere. Not oosphere, it will be oospore. So it will give rise to oospore. Oospore or zygote. 2n, this is n, this is n haploid, plus another independent fertilization event which implies the male gamete, which is again n, plus the secondary nucleus at the center of the embryo sac, which has a ploidy of 2n, which is known as triple fusion or vegetative fertilization. Gives rise to primary endosperm nucleus, that is 3n. Okay, these two events are known as, cumulatively known as, double fertilization. So these two events, they cumulatively are known as double fertilization. So this is the entire thing regarding the double fertilization. Now, what is the significance of double fertilization? That should also be understood in detail. The significance of double fertilization. Significance of double fertilization. Now what happens is double fertilization is found in angiosperms only. Number one thing is it is found in angiosperms only. Note it down. Angiosperms only. Next, in angiosperms, female gametophyte abruptly stops its growth. It doesn't undergo any more meiotic division after the 8th nucleate stage, means after the third mitotic division only. I said meiotic in the beginning, so it doesn't undergo any more mitotic divisions. Okay, after the 8th nucleate stage means 3 successive mitotic divisions. So further growth of the embryo sac occurs only and exclusively when the zygote has been formed. Okay, after the zygote has been formed. So after the 8th cell stage, I should be saying 8th nucleate stage, the embryo sac growth, it ceases. So the growth only happens after the formation of the zygote and the primary endosperm nucleus. So growth resumes for the embryo sac only after the formation of zygote and primary endosperm nucleus pen, which is 3N, this is 2N. Okay, they have been created by means of syngamy and triple fusion. So the triple fusion is the fusion, or the vegetative fertilization is the event that initiates the formation of pen and eventually forms the endosperm. Okay, the endosperm is formed only when it is needed. The need arises after fertilization because why? Because the endosperm provides nutrition. The endosperm provides nutrition for the simultaneously developing embryo. Nutrition for the simultaneously developing embryo. So this is what it does. Okay, if fertilization fails, always remember, if the fertilization event fails means if the syngamy or generative fertilization event, the formation of the zygote after the fusion of the oosphere and the male gamete, if that fails, then no endosperm is formed. Why? Because there will be no wastage of energy in the development of endosperm. So there is no such provision when we talk about gymnosperms. Okay, so that is the main way of conserving energy, and it's a feedback mechanism, you can say, in case of angiosperms. So this is therefore no wastage of energy on, specifically when you consider the higher order angiosperms. So this is what we should be thinking of when we are thinking of, or we are pondering upon, contemplating upon the double fertilization event. So this is what you should be thinking of, right? Then next is types of pollination. Okay, next event is types of pollination. When we talk about pollination, okay, so when the pollen grains are shed from anther, what happens is they are disseminated, okay, by various agencies. Some of them may be one means or another, and what they do is, their aim is what? The aim of the experiment or the aim of this specific event is to reach the stigma of a specific pistil, either of the same or another flower. That doesn't really matter, but you have to reach the stigma. Okay. Now, the transfer of pollen grain, the transfer of pollen grain from anther to stigma can be called as pollination. Called pollination. Now pollination ends when the pollen has reached the stigma. Now plants are said to be, you can say, self-pollinated when the pollen is transferred from an anther to a stigma of the same flower or the stigma of another flower on the same plant. Then it is a self-pollination event. It could be the same flower or the plant will remain same but the flower can change, doesn't matter, again it's the self-pollination event only. But the transfer of pollen from the anther of a flower to the stigma of a flower of belonging to completely another plant, you can say, is known as cross-pollination. Okay, so pollination can be divided into multiple types based on the table that I am about to give you. Draw this table properly, keep it with you. So if I talk about pollination, you could have self-pollination, and you can have cross-pollination or xenogamy. So autogamy, and I'll talk about xenogamy. So here you may have autogamy and heteronogamy. Autogamy, then heterogamy. Autogamy means same flower. Heterogamy means different flower of the same plant. Okay. Then you will be having xenogamy. Xenogamy may have abiotic agencies. Under abiotic agencies, you may have anemophily. You may have hydrophily. Okay. Under biotic agency, what you have is entomophily. Okay. We have chiropterophily. Chiropterophily. Okay. We have malacophily. We also have ornithophily. Okay. So this is the table that you should be knowing at all times regarding the pollination. Any question of pollination, if it is a simple question, you can get it done. You can answer that question specifically if you look at this table and you can comprehend this table properly. So keep this table with you. This is the best concise table regarding the pollination. If you don't know pollination, then even for 5 minutes if you go through this table, you can acquaint yourself and revise pollination in a very short period of time. So now if we talk about the autogamy, okay, self-pollination event, also known as homogamy sometimes, okay, because self-pollination usually takes place where? In the monoecious plants or in those plants bearing bisexual flowers, in which both male and female sex organs mature almost at the same time. It is again known as homogamy. So it can be further classified into, number one is autogamy. Now once we talk about autogamy, it is the transference of pollen grains. The transference of pollen grains from anther of a flower to the stigma of the same flower. Means that you cannot even change the flower here to the stigma of the same flower. Same flower. Okay. So that is evidently bisexual. Okay, the plant must be bisexual completely. So example, you have pea, you have wheat, okay, then you have rice, etc. Okay, you have multiple examples of such things. Next is gitonogamy. Okay, so number 2 is gitonogamy. So once we talk about gitonogamy, means when pollen from one flower are deposited on the you can say stigma of another flower, born on the same plant, This is known as gitonogamy because genetically it is self-fertilization. Okay. Now you have various things which can promote devices that can promote self-pollination. Okay. Number 1 is homogamy. So if you talk about devices that can promote self-fertilization, number 1 is homogamy. Now what is homogamy? So it is the condition in which I just had explained to you, I'll tell you again. It is the condition in which anthers and stigmas anthers and stigmas in bisexual flower attain maturity at the same time. Then we have cleistogamy. Very, very important is cleistogamy. Okay. So in cleistogamy what happens is the flowers never open to expose their self organs, okay, or their sex organs, as you were saying. The plants never expose to, or never open, I should be saying, never open to expose their sex organs, and the pollens fall on the stigma of the same flower. And the pollen grain fall on the stigma of the same flower. Now what happens is cleistogamy is followed by, you can say, geocarpy in plants like groundnut. Like fruits are formed in the soil. Okay, so some plants like viola, they produce both the types of flowers, that is chasmogamous and cleistogamous. Okay, so the cleistogamous flowers are invariably autogamous, as there is no chance of cross pollination. Okay, because they never expose their, you can say, sex organs. So pollinators are not required in such flowers at all. Very, very important, neat question. In case of cleistogamy, there is no need of pollination. Okay, so whereas chasmogamous flowers are with exposed anthers and stigma. So in a normal flower which will open its, you can say, anthers and stigma, complete autogamy will never occur. It is very, very rare. It can occur, but it is very, very rare. Okay, so it can then go for cross pollination. Also, it can accept, you can say, pollen grains from all types of agency. So I hope you are able to comprehend the lecture. If you had any doubts, we will be discussing those doubts specifically in the next class. Okay, so in the next class we will be talking about cross pollination and also the post fertilization event, okay, pertaining to the double fertilization and what happens, what are the events that happen after double fertilization, okay, pertaining to flowers. Okay, thanks a lot.